Welcome back to the bulk RNA-seq analysis pipeline within the Bridge Horizon platform. In part one of our demo series, we went through the bulk RNA-seq alignment pipeline, which generates the feature count tables from your raw FASTQs. One of the things that you can do with your feature count tables is use them for differential gene expression analyses. So in this part two of our demo series, I'm going to walk you through the differential gene expression pipeline within the Bridge Horizon platform. In order to launch this pipeline, you're going to click simply Launch Pipeline button within the DGE pipeline widget, and this will bring you to the sample sheet configuration page. To configure your samples, you click Samples Configured, which will bring you to the sample sheet. Now, my DGE sample sheet or DGE pipeline sample sheet has the following columns: sample ID. This is where you will define your replicates; condition column, which is where you will enter your conditions, whether it be control, treated, etc. In the feature count file column, you will enter or drag and drop your feature count file that could be generated, for instance, from the bulk RNA-seq analysis pipeline. And then this is where you'll enter your experiment ID. Now, my DGE files or feature count tables are contained in this DGE test folder. One really nice feature of the sample sheet configuration page is you can drag and drop your feature count files. So as you can see, I've selected 6 files. I'm just going to drag them and drop them into the feature count file column. The next thing I'm going to do is label my condition. So all my samples with the T annotation are my treated samples. I'm missing one. I'm missing T3. So I'm just going to drag and drop it here. All the samples with the T annotation are treated samples, and then all the samples with all the feature count files with the C annotations are my control samples. And then T2 would be replicate number 2 for my treated, and C2, for instance, would be replicate number 2 for my control condition. So at this point, I'm going to label my conditions. So this would be treated. And then instead of typing treated for all my replicates, I can simply copy and paste. And this is the third replicate for my treated condition. I'm going to enter control over here and then copy and then paste. At this point, I'm going to name my sample IDs. So this is T2, which is my second replicate from the treated condition. So I'm going to simply call it T2. This is T1. This is C3, C2, C1, and then T3. And lastly, I'm going to give this an experiment ID. Since these are all from the same experiment, I'm just going to call it experiment 1. And then I'm going to copy and paste. And just like that, I have very simply configured the sample sheet in order to execute the pipeline. Once you're happy with your sample sheet, you can click Done, which will bring you back to the sample sheet configuration page. And as you can see, it shows that I have 6 samples configured. You can also learn more about the pipeline by maneuvering to the help page or in the about section, which will launch the full documentation on the differential gene expression pipeline, as well as the appropriate references. This page is very helpful if you're trying to learn our workflow for your publication, grants, or simply to better understand how the pipeline works. So once you have familiarized yourself with the pipeline and you have your sample sheet filled out, you're going to click Next, which will bring you to the pipeline tier. Now, the DGE pipeline has been coded in a way that it works with a single pipeline tier. So regardless of how many experiments you have, how many feature count files you have, or how many groups you're comparing, the single DGE tier is intelligently designed to work with all of them. So you're going to select your tier and then click Next. You can see also within the parameter section, the files that I have selected for comparison. Click More to view all the feature count tables, my pipeline tier, and the estimated time of completion. At this point, I'm going to give it a run name. So I'm going to call it, these are zebrafish DGE or feature count tables that I'm using for differential gene expression analysis. So I'm just going to say zebrafish DGE and then launch pipeline. And as you can see, the pipeline is currently running. We will return once the pipeline has completed running. And then I'll walk you through the outputs. Okay, everyone, as you can see that the pipeline has finished running successfully. In order to visualize the outputs from the pipeline, you're going to simply go into the submission outputs and then click Explore. And this will take you to the pipeline outputs. Now, the folders have been designed or written in such a way so that you can easily understand not only when the run was completed, but also which sample groups you were comparing. And the DGE pipeline works in a way that if, let's say, you had 3 different conditions, then you would have 3 separate folders over here indicating 3 separate pairwise comparisons. So, and once again, if you had multiple experiments, this page over here will be populated by as many pairwise comparisons that you could possibly do with the number of conditions that you specify within the sample sheet. In this instance, since we only had 2 conditions, you could only do a single pairwise comparison. and therefore you only have a single folder that contains your DGE analysis. If you click this folder, this will take you to your metadata, which contains some very basic details on the version of R or the DSeq2 that we use within the pipeline, your condition 1, condition 2, the project ID, the total number of genes, and the number of significant genes that pass your p-adjusted threshold. Your results are contained within the DGE folder. So if you go and maneuver into this folder, you'll find that you have a variety of different outputs. The differential gene expression analysis is contained within this folder or this file over here, which is annotated as DGE results. And then the one thing that we have done is that we compute MD5s for all the relevant files, important files, so that if you're downloading these, you can run an MD5 sum on your end to ensure file integrity. Now, in addition to the DGE results, we also output a normalized count file. So as you probably know, the DSeq2 algorithm performs normalization before it does differential gene expression analyses and the statistical analyses. A lot of people like to have a normalized count as a separate output so that you are able to generate visualization using R or Python scripts on your own end. And this file, which contains your normalized counts, can be used for that. All the plots and the visualization that are generated from this pipeline are contained within this plot subfolder. As you can see, we generate a variety of different visualizations. We have performed dimensionality reduction within the DGE pipeline, and that can be visualized using a scatter plot that is contained within your PCA 2D or PCA 3D folders. You also get a heat map looking at the top 100 genes as default, but you can obviously adjust that within the advanced setting when running the pipeline. You also get a volcano plot and then a MA plot, which allows you to visualize how the data was normalized. So this has been a demo of the DGE analysis pipeline within the Bridge Horizon platform. I hope you enjoyed this demo. If you have any questions, please let me know. Thank you.